question is name and describe the technique that helps in separating the DNA fragments formed by the use of restriction endonucleases. So the technique that helps in separating the DNA fragments formed by the use of restriction endonuclease is DNA fingerprinting. So DNA fingerprinting works as follows. So if we see the steps of DNA fingerprinting, the first step is the extraction of DNA from the given sample. So the DNA is extracted from the nuclei of whatever evidence is available. Say for example from WBCs in case of blood sample, or from hair follicle cells that cling to the roots of hair that have fallen or have been pulled out, or from spermatozoans in semen sample. Now the next step is amplification of DNA molecules by using polymerase chain reactions. So if the content of DNA is limited, then DNA can be amplified by making many copies of it by using PCR or polymerase chain reaction. Now the next step is the DNA sample is digested by a restriction enzyme which cuts the DNA into fragments at specific sites. So now these fragments are separated by gel electrophoresis setup containing agarose polymer gel. So after this gel electrophoresis, so these double-stranded DNA is then split into single-stranded DNA using alkaline chemicals. So these separated DNA sequences are then transferred to a nylon or nitrocellulose sheet placed over the gel. So this is called Southern blotting. So basically Southern blotting is a technique where the DNA band pattern in the gel is transferred to the nylon membrane. Next, now this nylon sheet is then immersed in a bathe and probes or markers that are radioactive synthetic DNA segments of known sequences are added. So these probes target a specific nucleotide sequences which is complementary to VNTR sequences. So VNTR is nothing but variable number tandem repeat sequences and then hybridizes them. So the final step of DNA fingerprinting is X-ray film that is exposed to the nylon sheet containing radioactive probes. So you can see in this figure, the X-ray film is placed next to the membrane to detect the radioactive pattern. So dark bands develop at the probe sites which look something like barcodes which are used to identify items at the grocery stores. So the degree of variation is so high that every individual, with the exception of identical twins, so this is exceptional in case of identical twins, and whereas so the degree of variation is very high that every individual produces a unique band pattern, as every individual has a unique set of ordinary fingerprinting. So that was about the DNA fingerprint.